In this video, we will discuss about another example of sting agonist enhance the killing of human AMC cell lines when they are targeted using this BH3 mimetics. So first, why they have selected this AMC cell lines? Because this human AMC cell lines has high expression of the sting protein. So in order to validate whether they have the expression of sting proteins or not, they did the Western blot in different panels of this human AMC cell lines, and the results are shown in the panel A, where we can see that the sting expressions are there in all the AMC cell lines that they have tested in this Western blot. Thereafter, they check the effect of sting alone on these AMC cell lines by 3 different reagents that can trigger sting pathways, and they can see the cell death occurring in all the 3 cases. That is shown in this panel B, where they have used MOM13, MV14 and THP1 cell lines, and the reagents that they use to trigger sting pathways is ADU S100, MSA2 and diabzole. And in order to check whether it is triggering this apoptosis genes, they check the gene factors that are upregulated by apoptosis, such as NOXA, PUMA, BIM, IFN, beta. All of them are upregulated when this diabzole is used, compared to the control where only DMSO is used. And thereafter, what they did is they check the effect of combined therapy. So far, they are only checking the effect of the sting signaling pathways activations. After some time, they check the effect of combined therapies on these AMC cell lines, such as MOM13, MV14, THP1, and what they have found out is that there is a significant decrease in the live cells when the combined therapies are used for both the cases, where in the control AMC cell lines and in the case where TP53 has been knocked out, as TP53 functions will be taken care by this activation of sting pathways. So they see the same results in both the in all the 3 cell lines, that is MOM13, MV411 and THP1. We can see that significant decrease in the live cells compared to the other case. And after validating that the combined therapies works well for the human AMC cell lines in vitro, they wanted to check whether this combined therapy works in vivo or not. So they took this NSG mice and treated them with this ventolex to trigger BH3 mimetics, and they triggered or inject this diabzole reagent to trigger sting pathways, and they use different combinations of this to check which combinations works better. And when they did using this MOM13 control cells, what they have find out is that the individual therapies, that is when they use this ventolex, they see some increase in the survival populations, and when they use only the diabzoles, they see some more increase in the survival populations. But the maximum increase in the survival populations occurs when they use the combined therapies, and these things they have observed for both the control cells and also for this TP53 knockout. And these results are shown here for this MOM13 cells, MOM13 cells that are injected into this NSG mice. So in one case, they inject the control mice and did the experiment with the control with the BH3 mimetics with the sting signaling pathways alone, and thereafter, and also this combined therapy effect. And same things they did it when in the MOM13 cell lines with this TP53 knockout. So in this case, TP53 knockout MLM cell lines injected into the injected into the mice, not patients, into the mice, and then following treatment are done. The map is shown in this panel E, where we can see that at what time this ventriculox is injected and at what time diazole is injected. And the overall outcomes comes out that the combined therapies, that is sting when used along with this BH3 mimetics, has a higher percentage of survival populations or higher percentage of survival mice compared to the monotherapies or non-treated or mice that are not treated with any therapies. So this further substantiate their points, that further substantiate their combined therapy efficiency in the case where sting proteins are expressed in the cancer cell lines. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.